We're on chapter 4. Okay, can someone please confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? What happened here? A sticky mess on my desk. If it's on my jacket. All right, I'm not hearing any questions. Let me share my screen here. And I didn't think I opened lecture 4. So let's open it. Sharing my screen. Should be able to see my screen now. I have that we had talked about the membrane of gram-negative and gram-positive cells. Any question about any of that? All right. Let's see. Today is January 29th, and we're supposed to be working on chapter 4. In the lab today, I'm going to explain the unknown project. Nope, I take that back. There is no lab today. You're supposed to read the unknown project, and I'll discuss it in the lab next week. Any questions about any of that? So I will be showing up in the lab only to answer questions for about 15 minutes. After the first 15 minutes, or the last question gets answered, I will log off. And I'll be there for at least 15 minutes. Longer if there's more questions, but usually there aren't too many questions. And so I'm usually there for only 15 minutes. You do not need to show up in the lab unless you have questions or you want to listen to the questions. But that's what we'll be doing in the lab today. Any question about any of that? Sorry, I got something sticky on my table here. I think it moved on my jacket.
So we're not going to discuss the unknown project until next week because I read it last night and it was kind of confusing. Like I understood if I was in like an in-person class how uh how we would do that in the lab, but like I wasn't seeing like any material to like base my game plan off of, you know?
I will talk about it next week.
Okay.
Okay. It's especially designed to be done all online, so you're not coming into the lab to do the unknown project. All right. Let's go to chapter 4. Any other questions, I guess? So the gram-negative outer membrane does contain numerous molecules like the lipopolysaccharides, including lipid A, lipoproteins, phospholipids, which you find in all membranes. The gram-negative outer membrane can protect the cell from the complement proteins, which you haven't really talked about much, but they're immune system proteins that can attack invaders like bacteria. But the outer membrane can also protect the gram-negative cell from certain antibiotics. The O polysaccharide antigen can protect the cell from phagocytes, so red blood cells cannot consume the bacteria if it has a capsule made up partly of O polysaccharide antigen. That's the O polysaccharide antigen is the polysaccharide attached to the lipid A. And the O polysaccharide antigen actually gives the E. coli antigenic characteristics like E. coli 0157H7. The 0157 is an antigen from the O polysaccharide, and this E. coli is actually a human pathogen. You do not want this. You'll get gastroenteritis if you get this E. coli. This is a different coli from the normal E. coli, which is in everyone's gut. This E. coli is a human pathogen. Remember that lipid A is an endotoxin, and the gram-negative outer membrane does have porin proteins that form channels through the outer membrane, and that allows important molecules to enter the cell or leave the cell. So if it can't get through the outer membrane, it has to move through the porin protein. That's a little bit different than the cell membrane. We use transport proteins to move one specific protein or other molecule through the cell membrane. And that means there's a different transport protein for glucose than there is for fructose. But both glucose and fructose move through the porin protein. Any question about any of that? All right, taking a look at the gram-positive cell wall and how it differs from the gram-negative cell wall. The main difference is the gram-positive cell wall has a thick peptidoglycan layer. It's a rigid structure. The gram-negative cell wall has a thinner peptidoglycan layer, something like between 3 and 20 layers. It is not as rigid. There is teichoic acid in the gram-positive cell wall. The teichoic acid helps hold the gram-positive, meaning the peptidoglycan layers, together. There is no teichoic acid in the gram-negative cell wall. There is no outer membrane in the gram-positive cell wall. There is an outer membrane in the gram-negative cell wall, and that outer membrane contains lipid A, an endotoxin. Any question about any of that? So right off the bat, you should know that whenever you get infected with a gram-negative cell, you're exposed to an endotoxin, lipid A. That is not the case with gram-positive cell walls. To be exposed to a toxin, the bacteria cell will have to make it. A gram-negative cell wall that has a cell wall, that's a little bit of an oxymoron, will always have lipid A in it. So you'll always be exposed to an endotoxin if the gram-negative cell has a cell wall. Any question about any of that? Sorry, I'm still running into that sticky stuff. Wish I would have found it before class, then I could have wiped my desk down. All right. The gram stain mechanism happens because of the difference in the cell walls. The crystal violet iodine complex forms in all cells. In a gram-positive cell, the crystal violet iodine complex does not leave the thick peptidoglycan layer. The gram-negative cell, the alcohol of the alcohol wash will dissolve the outer membrane, and the crystal violet iodine wash will wash out of the thin peptidoglycan layer. And then safranin will then stain the cell pink. Why gram-negative cells stain pink and gram-positive cells stain purple or purplish? Any question about any of that? So looking at a summary of the gram-positive and gram-negative cells, let me blow this up so you can see it better. The gram-positive cell on the left column, the gram-negative on the right. The gram reaction obviously stains purple, stains pink from the safranin. The peptidoglycan layer is thick in the gram-positive cell, thin in the gram-negative cell. Teichoic acid present in gram-positive cells, absent in gram-negative. You don't really need to know about the periplasmic space. That's the area of the cell where in the gram-negative cell, it's the region between the outer membrane and the cell membrane where the peptidoglycan layer is, And that's only found in the gram-negative cell because, of course, in the gram-positive cell, there is no outer membrane. The outer membrane absent in a gram-positive cell, present in a gram-negative cell. The lipopolysaccharide, virtually none in a gram-positive cell, and is present in high amounts in the gram-negative cell. It's the endotoxin and the attaching old polysaccharide attached to the endotoxin. You don't need to know any of the lower things. Any question about the difference between gram-positive and gram-negative cells? All right, not hearing anything. So we've talked about the gram-negative cells and the gram-positive cells and talked about their differences. Let me state there are some cells with atypical cell walls. And I actually should move this to the first one up here. Archaea is an atypical cell wall because they're not bacteria. Everything we've talked about is bacteria. The archaea are a different domain and they have their own cell walls. Instead of made of peptidoglycan, it's made of pseudoaminine, another disaccharide. Some of the archaea lack a cell wall. As I mentioned, some of the bacteria do. So the archaea have different cell walls. Any question about that? All right. In the bacteria, there are two families which have atypical cell walls. First, there's the mycoplasmas. I've actually talked about them before. Let me get that out of the way. Can't get it out of the way. Let me move on and I'll come back. Hopefully that'll get out of the way. Yep, it did. All right. The mycoplasmas are bacteria, but they lack a cell wall. They have sterols in the plasma membrane, similar to the sterol in animal cells. If you remember, in animal cells, we have cholesterol in our cell membrane to give the cell membrane strength. Well, the mycoplasmas have another sterol in their cell membrane to give the cell strength. And that's because they lack a cell wall. They stain gram-negative because they don't have peptidoglycan in their cell wall. They don't have a cell wall. Any question about the mycoplasmas? All right, the other family of bacteria which is different is the Mycobacterium. They have acid fast staining properties because they have a lot of mycolic acid in their cell wall. This mycolic acid makes the water-soluble molecules difficult to get inside the cell because the water-soluble molecules don't go through the mycolic acid. And then the mycolic acid also prevents water inside the cell from leaving the cell, giving the cell an acid fast staining property because of all the mycolic acid. It allows the acid fast stain to come in and then it gets stuck in the cells because of the mycolic acid. All right, any question about these atypical cell walls? You can damage prokaryote cells, and once you damage the prokaryote cell wall, the cell will be susceptible to osmotic lysis. That's actually how penicillin kills cells. It inhibits the peptide bridges from forming in the peptidoglycan, the crosslinks. That'll create a hole in the cell wall, and then the prokaryotic cell will be susceptible to osmotic lysis. You can also treat the cell with a lysozyme. This is an enzyme which you can actually find in your skin. It digests the disaccharide in the peptidoglycan, removing the cell wall from the bacteria. You don't need to know there's different terms for a wall-less gram-positive cell and a wall-less gram-negative cell. You don't need to know those terms. Just know that lysozyme can digest the cell wall of the bacteria, and then the cell will be susceptible to osmotic lysis. Water will move into the cell. There's no longer a functional cell wall to protect the cell membrane, and then the cell will eventually burst from water moving into the cell. Any question about any of that? Of course, that means the cell has to be in a environment where there's more water outside the cell than inside the cell. Bacteria often find themselves in environments like that, such as the bacteria in your blood. The blood has more water in it than the bacteria cell has, at least most bacteria, and then water will move in to the cell with the osmosis, and because the cell has a non-functional cell wall from either penicillin or lysozyme, the water will move in and eventually kill the cell. Any question about any of that? That's why penicillin is so effective, by the way. All right, gram-negative bacteria are not as susceptible to gram-positives to penicillin G because of the outer membrane. It doesn't let the penicillin G get inside the cell and then do damage. However, that doesn't mean the gram-negative cells are not susceptible to different antibiotics, like ampicillin, the third member of the penicillin family, can kill both gram-negatives and gram-positives. And then the gram-negative bacteria are also susceptible to antibiotics that can penetrate the outer membrane. Streptomycin is such an antibiotic. Streptomycin is a lipid-soluble antibiotic, and it can get right through the outer membrane of a gram-negative cell. Any question about any of that? All right, let's talk a little bit about the plasma membrane. It is a phospholipid bilayer where the phospholipids arrange in two layers. One, the inner side where the head is facing inside, the tails face inside the bilayer, and then there will be another layer of phospholipids with the head facing outside the cell, and then the tails facing inside the cell. Let me blow this up. So there we have the cell membrane, plasma membrane, and you can see the phospholipids facing inside the cell where the head is facing the water or the cytoplasm, and then the head of the other lipid bilayer is facing outside the cell because outside the cell is mostly water. Think of this as a red blood cell. The blood is mostly water, and so the head is facing mostly water. It doesn't matter what we're in because even in bone, the majority of the bone is water. So outside the cell, there's mostly water. And then the fatty acid tails face inland, inside, because they like to be among themselves or among lipids, and they do not like to be around water. The plasma membrane has other molecules inside of it, including proteins. There are three different types of proteins we can find in the cell membrane: peripheral proteins, integral proteins, and transmembrane proteins. Integral proteins and transmembrane proteins are proteins that span the entire membrane. Like this looks like a channel protein, allowing things to come into and out of the cell, and it's an integral protein. A transport protein would be a transmembrane protein, and it'll pump molecules either inside or outside the cell. My point is it spanning the membrane. Here we have a peripheral protein. A peripheral protein is one that is inside the cell membrane, and the majority of the protein is on one side of the membrane or on the other side of the membrane. Any question about any of that? We use the fluid mosaic model to describe the cell membrane. The fluid mosaic model states that the membrane is a mosaic of phospholipids and proteins, and the membrane is as fluid as olive oil. Any questions about any of that? All right, think about that. You're seeing my hand here. It's held together only by the cell membrane, and the cell membrane is a fluid as fluid as olive oil, holding my hand together. Every time I think about that, I always am amazed by that. Any
Really?
question? Uh-huh. Was there a question? All right. Obviously, we have the skin too, but which is dead cells. But my point is the animal cells are held together by something as fluid as olive oil. The cell membrane is as fluid as olive oil. This means that many, not all, but many of the proteins are free to move about in the cell membrane. And those of you who have had AMP and you know that a receptor binds to a protein, they usually come move together and then they are brought inside the cell as a group, meaning the proteins, many of them, are free to move about in the cell membrane. Not all of them, but many of them are. The proteins which are not free to move about in the cell membrane are ones that are anchored either to an external source, like the extracellular membrane, or the ones that are anchored to the intracellular membrane. Like this protein here could be anchored to the intracellular matrix, which is a matrix of proteins running throughout the cytoplasm of the cell, and it just tends to give the cell shape, especially in eukaryote animal cells, but in bacteria, the shape is more from a cell wall. But my point is, if this cell is anchored, this protein is anchored to the intracellular matrix, then this protein is not free to move about in the membrane. And the same with this protein. If it is anchored to the extracellular matrix, it is not free to move about in the membrane. Now, for eukaryotes, I can explain the extracellular matrix. Let's say this is a cell in the bone marrow, and this protein is literally anchored to the matrix of the bone. This protein then will not move about in the cell membrane. I'm not sure what the extracellular matrix would be in a bacteria because bacteria tend to live as single cells. So I'm not sure what the extracellular matrix would be, but the intracellular matrix I've explained. Any question about any of that? All right, the phospholipids are free to move about in the membrane, and they can move laterally, or I mean rotate all the time. So this can just rotate right in place here, spinning around. But it can also move that way. The thing is, it's got this in the way, so that has to move out of the way, and then that one can move over. That's actually how water can move through the cell membrane. Water being outside the cell simply waits for a phospholipid to move over, and then the water occupies the space. Come on, mouse. Where this phospholipid used to be, it can't get through this uh, fatty acid portion of the lipid portion of the bilayer, but the water just waits there until this phospholipid moves over, and then the water moves through the membrane this way. So that's one way that water can move into the cells. It's not a very efficient method for moving water into the cell, and many cells have aquaporins. These are transport proteins that transport water, well, usually into the cell. Like the roots have aquaporins on them, and they can transport water into the cell and then make water move inside the cell very rapidly because the water moving through the cell membrane, it does move inside the cell, but it moves very slowly, meaning it has to wait for this phospholipid to move over, then it occupies this space, and then it has to wait for this one to move over before it can move into the cell. Any question about any of that? All right, the plasma membrane shows selective permeability, allowing the passage of some molecules, but not others. No large molecule can move across the cell membrane unless there is a transport protein to move the molecule across the cell membrane. Any question about any of that? So let's talk about a teenager making hormones, dumping out hormones in the blood, the hormone being absorbed by a cell. It has to have a transport protein to bring that hormone into the cell. Any question about any of that? Ions either do not move across the lipid bilayer of a cell membrane or they move very slowly. Lipid soluble molecules can move across the lipid bilayer. But think about it, a cell is most of the time in blood or environments similar to blood. There's not a lot of lipids in the blood. There, because the blood is mostly water, but there are some. You know that they test your blood for cholesterol level and what is it? Lipopolysaccharides, or trying to remember what the thing is called in the high density cholesterol and low density cholesterol. Those are lipid soluble molecules. They are inside the blood, although they have to be um, surrounded by molecules which will allow the lipid to be inside the blood. But they can move right through the cell membrane, is my point, because it's a lipid soluble molecule. Smaller molecules can move across the cell membrane. Like I mentioned, water can. It doesn't move very quickly. That's why we have aquaporins to move water across the cell membrane quickly. But then there's two other molecules that easily move across the cell membrane. That's oxygen and carbon dioxide. Oxygen easily diffuses into the cell across the cell membrane, and carbon dioxide easily diffuses across the cell membrane to leave the cell. Any question about any of that? Oxygen and carbon dioxide are both nonpolar molecules, and small nonpolar molecules can easily cross the cell membrane. The main two being oxygen and carbon dioxide. All right, let's talk about some miscellaneous things with the plasma membrane. The plasma membrane of bacteria as well as archaea contains the enzymes for ATP production. We'll talk about that when we're talking about aerobic respiration, but the ATP enzymes are actually in the cell membrane. That's not true of eukaryote cells. Eukaryotes have the ATP producing enzymes inside the inner membrane of the mitochondria. Once again, it's in the membrane, it's just not in the cell membrane. Bacteria and archaea also have the photosynthetic pigments found in foldings of the cell membrane. Let me blow the slide up. We call these foldings of the cell membrane either chromatophores or thylakoids. And here we have the cell membrane running along there, but it's not a single thing like that. The cell membrane, oops, let me go back. The cell membrane, let me blow this up to make it a little clearer. Runs like this outside the cell wall and then folds around like that. Move over to here, folds around like that. Moves over to here, folds around like that. And inside these chromatophores or thylakoids, we have the enzymes and the photosynthetic pigments. Any question about any of that? All right. right, damage to the plasma membrane will cause leakage of the cell contents. Damage can occur with alcohols, quaternary ammonium compounds, or the antibiotic like polymyxin B, putting a hole in the cell membrane. If a hole in the cell membrane, the cytoplasm can leak out, and that will kill the cell. Any question about any of that? All right, movement of materials across the cell membrane is critical to cells. There are two ways that materials can move across the cell membrane. There's a passive process and an active process. The passive process, molecules move down their concentration gradient, meaning if molecules are higher outside the cell, concentration is higher outside the cell, they can move inside the cell down the concentration gradient. Simple diffusion, actually, let me state that passive process can happen in three different ways. Simple diffusion, where the solid simply moves down the concentration gradient. An example of that would be oxygen, higher outside the cell than inside the cell. It simply diffuses into the cell. Diffusion can only happen by a solute, a molecule dissolved inside the solvent. Facilitated diffusion happens the same. It's a passive movement where the molecule moves from areas of higher concentration to areas of lower concentration. It moves across a membrane, like the cell membrane, and it has to have a protein to facilitate this diffusion, what's called facilitated diffusion, where a channel protein can allow a molecule to move from areas of higher concentration to areas of lower concentration. Once again, that molecule must be a solute. Osmosis is the movement of water across a membrane from areas of high concentration to areas of low concentration. Water is a solvent, and osmosis is specifically the movement of water across a membrane from areas of high concentration to areas of low concentration. Any question about that? So osmosis happens by a solvent, and it's always defined as water because water is critical to living cells. We don't have cells in alcohol or olive oil, for example. Although if you did, would that be called osmosis? No, because water is defined as osmosis is defined as water. I don't know what it would be if olive oil were to move inside the cell. I have to look that up. Any questions about passive processes? All right. An active process is where the molecule moves against its concentration gradient, where the molecule moves inside the cell, but the molecule is higher inside the cell than outside the cell. Active process always requires an active transport protein pumping the molecule against its concentration gradient. This requires energy to move the molecule against its concentration gradient, and it requires one ATP molecule to move one molecule against its concentration gradient. So if the cell wants a molecule like glucose inside the cell, and yet the glucose is lower outside the cell than inside the cell, it can move the glucose into the cell by active transport, spending one ATP molecule to move one glucose into the cell against its concentration gradient. Any question about any of that? All right, let's talk a little bit more in detail about passive processes. Remember, there are three: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is just the movement of a solid from an area of high concentration to an area of low concentration. Like when we put a drop of dye in a beaker of water, the molecules of the dye will move by simple diffusion from areas of high concentration to areas of low concentration, shown here. Simple diffusion does not require a membrane, like the cell membrane. But simple diffusion can occur across the membrane, and we have oxygen and carbon dioxide moving into the cell if it's oxygen by simple diffusion, and then carbon dioxide moving out of the cell by simple diffusion. And the molecules are just moving down their concentration gradient from areas of high concentration to areas of low concentration. Any question about any of that? All right, facilitated diffusion is similar to simple diffusion, except the molecule is moving from areas of high concentration to areas of low concentration across a membrane. How that happens is with a protein facilitating the diffusion. Usually it's called a channel protein. Here it's called a transporter protein. Don't get it confused with a transport protein. Glucose is not a good example, and I'll explain why in a minute. Fructose is a better example. Fructose only moves into the cell by facilitated diffusion. So when you're eating fruit and you have a lot of fructose in your gut, it will move into your intestinal cells, the cells lining the intestines, by facilitated diffusion. That means once the concentration of the fructose outside the cell equals the concentration of fructose inside the cell, there will be no net movement of fructose into the cell. So fructose will never be fully digested. Any question about any of that? That's different with glucose. Glucose, when it's higher outside the cell than inside the cell, will move into the cell by facilitated diffusion until the concentration of the glucose outside the cell equals the concentration of the glucose inside the cell. Then facilitated diffusion stops and the protein changes its activity from becoming a facilitated protein, meaning a channel protein. It literally changes into an active transport protein, and then it will pump all the remaining glucose that is outside the cell into the cell against the glucose's concentration gradient. And to move one glucose into the cell by active transport, it will require an expenditure of ATP. So one glucose will move into the cell by active transport through an active transport protein, and that will cost the cell 1 ATP. This means that all the glucose you eat, all of it will be digested. And that protein literally changes its activity and its shape depending on whether the glucose is higher outside the cell or lower outside the cell. If it's higher, the protein will be acting as a facilitated diffusion, and then the glucose can move into the cell without the cell spending energy. But once the glucose is equal outside the cell and inside the cell, this protein will change its shape and activity and become an active transport protein. Any question about any of that? All right. That's why glucose is not a good example to show facilitated diffusion. In both simple and facilitated diffusion, the solid molecules are transported. The transport happens down a concentration gradient. from areas of high concentration to areas of low concentration. Osmosis is the net movement of water. Water is a solvent, not a solute, and this has to happen across a selectively permeable membrane. So osmosis does not happen in the absence of a membrane. That would just be simple diffusion if it's the absence of a membrane. But if water moves into the cell across the membrane, it will happen by osmosis, where the water molecules will move from areas of high concentration to areas of low concentration. Any question about any of that? Now realize when the water is higher outside the cell, that means the solute concentration outside the cell will be lower, because the solute concentration will be the opposite of the water. This is important because when we talk about the tonicity, the water environment the cell is in, the tonicity is defined by the solute concentration, not by the water concentration, even though it's the water that will be moving by osmosis. Any question about any of that? All right. A cell can find itself in three kinds of osmotic solutions called the tonicity. The cell can be in an isotonic solution, a hypotonic solution, or a hypertonic solution. You should note that each of these are relative terms, so it would be correct to state that the solution outside the cell is hypertonic relative to the solution inside the cell. Any question about any of that? Isotonic solution is the easiest one to explain, and that is where the solute concentration outside the cell is equal to the solute concentration inside the cell. That means there will be no net movement of water into the cell. The cells will be happy. They'll stay alive. They don't have water moving into the cell. They don't have water moving out of the cell, which could kill the cell. A cell in your blood, for example, like your red blood cells, are inside an isotonic solution. What do I mean by there's no net movement of water into the cell? Anyone have a guess?
It's
I'll give you any.
hydrophobic.
What?
Is it hydrophobic?
Um, no, water can move through the
It
cell
means
membrane. I mentioned that before. It slowly moves through. It waits for the phospholipid to move over, and then it moves into that first layer of phospholipid, and it just waits for the second phospholipid to move over, and then the water can move into the cell. So it can move into the cell, but what do I mean by there's no net movement of water? If one moves into the cell, if there's no net movement of water, what happens? Not getting an answer, so I'll answer it. It means there's, for every water molecule that moves into the cell, one water molecule has to move out, meaning there's no net movement of water into the cell. Doesn't mean water doesn't ever move into the cell. It's just when one moves in, one has to move out. That's what I mean by no net movement of water. For our purposes, we can just say that when the water is isotonic, in simple terms, there's no movement of water either into or out of the cell because there's no net movement into or out of the cell. In a hypotonic solution, the solute concentration outside the cell is lower than the solute concentration inside the cell. That means there's more water outside the cell than water inside the cell. That means water will move into the cell down its concentration gradient. That will cause the cell membrane to expand. If the cell has no cell wall or a cell wall with a hole in it, the cell membrane will begin protruding outside the hole or just protrude all the way if there's no cell membrane. Eventually, enough water will move into the cell that will pop the cell membrane. The cell will burst, the cytoplasm will leak out, and that would kill the cell. So in a hypotonic solution, if the cell lacks an intact functional cell wall, water will move into the cell until the cell dies by osmotic lysis. And as I mentioned, that's how lysozyme and penicillin G actually damage cells. Most bacteria live in an environment that's hypotonic, and it's their cell wall that protects them. Because with this intact cell wall, the cell membrane can only expand up to the size of the cell wall. Once they are touching each other, the cell membrane cannot expand any further. That will limit water moving into the cell because there's no space for the water to move into the cell. So no more water will move into the cell, and that will prevent the cell from bursting by osmotic lysis. And that's how the cell wall protects many bacteria. Probably most bacteria, but I'm just going to say many bacteria. Any question about any of that? All right, the third tonicity is a hypertonic solution where there is higher concentration of solutes outside the cell than inside the cell. That means there's more water inside the cell than outside the cell, and water will leave the cell by osmosis. That will cause shrinking of the plasma membrane away from the cell wall. We call that plasmolysis because the cell generally will die in a hypertonic solution. It does depend on what cell we're talking about, like endospores can survive for centuries in a hypertonic solution. I think that's really not a living cell, it's a frozen cell. But they've obtained endospores from the Egyptian mummies dating back at least 8,000 years, where they've gotten the endospore to germinate after 8,000 years. So the endospore can live a long time. Most endospores don't live 8,000 years, by the way, but some do. And then another cell like E. coli is very sensitive to plasmolysis. You put E. coli in a hypertonic solution like E. coli falling into a bottle of honey or a bottle of molasses. The water will leave the E. coli and then that cell will die quickly within a day. So the length of survival will depend on the cell, like Staphylococcus species can survive longer than many other cells like E. coli. But eventually a Staphylococcus aureus falling into a bottle of molasses, for example, it would eventually die. Eventually even an endospore would die, but as I stated, some endospores can survive for thousands of years in a hypertonic solution. So generally, a hypertonic solution does lead to cell death, but it does depend on the cell. And the two things which allow the cell to live in a hypertonic solution is if the cell has an endospore or the cell has a lot of mycolic acid, like mycobacterium falling into a bottle of molasses. That mycobacterium can survive in the bottle of molasses for a long time. It will not reproduce, but it can survive. I don't know how long it can survive, but it can survive for a longer time than a lot longer time than E. coli, for example. Any question about any of that? All right. Active transport is the movement of material against its concentration gradient. A passive process is okay when the concentration of a substance required by the cell is higher outside the cell than inside the cell. But what about a cell in an environment like the open ocean or the desert, like the Sahara Desert? The concentration of the molecules outside the cell will be much lower than the concentration inside the cell. And remember, the open ocean is a desert on the planet Earth, similar to this type of a desert. What's a cell to do when it wants to move something from areas of low concentration to an area of a higher concentration? It uses active transport, moving a molecule against its concentration gradient. This requires energy. It takes work to move the molecule against its concentration gradient, and the cell will obtain that energy from ATP. The cell has to have an active transport protein to pump the molecule against its concentration gradient. Generally, these active transport proteins are molecule specific, like we have an active transport protein to pump the amino acid lysine inside the cell, and it's a different active transport protein to pump the amino acid arginine inside the cell. Any question about any of that? The active transport protein is not entirely molecule specific. As you know, we have some drugs which mimic the ability of the substrate to bind to the active transport protein. And then that drug will tie up the active transport protein so the real molecule can't use it. So we have some drugs that do, but generally beyond those drugs, an active transport protein only moves one molecule against its concentration gradient and through a cell membrane, by the way. All right, any questions about active transport proteins and active transport in general? We do have a different mechanism of active transport in prokaryote cells called group translocation. Group translocation is only found in prokaryote cells, meaning bacteria, and archaea. It is not found in eukaryotes. A group translocation does require a transport protein. It does require a high energy phosphate compound, but it's not usually ATP. It's some other high energy phosphate compound like PEP. You don't need to know about PEP, but it just says another high energy phosphate compound. And then when the molecule is moved inside the cell by group translocation, let's say glucose is moved into the cell by group translocation, the transported molecule is chemically altered. For example, when glucose is moved into the cell by group translocation, once the glucose is moved into the cell, it is converted into glucose 6-phosphate. So when the molecule starts its movement, it's glucose, but when it ends its movement, it is glucose 6-phosphate. PEP usually transfers the phosphate molecule to the glucose or the sugar, and the PEP also gives the energy that the protein that engages in group translocation needs to move the molecule against its concentration gradient. The alteration of the molecule moved by group translocation is thought to help prevent the escape of the molecule from the cell and then allow the cell to accumulate substances more easily than if the molecule were to be transported by active transport alone. Let me see if I can show you this. Actually use paint. Is it going to open? There it is, just taking its time. Let's make this a little bigger. All right, so we have a cell. We'll make it right here, moving glucose into the cell. I'm not getting that to work. Why is that? Paint stopped working, that's why. Can anyone hear me?
Yes.
Please confirm you can hear me. For some reason, paint stopped working and I was afraid my whole computer stopped working, but it's not the case. Let's try paint again. That's not even closing down. All right, there we go. So there's the cell. We're moving glucose into the cell. There, it's working. So let's say we have 20 glucose molecules outside the cell. If we were to use active transport alone, the 19th molecule to move, actually the 20th molecule to move, you'd have one molecule outside the cell moving into a cell that has 19 molecules. So the concentration gradient is 1 over 19, which is a very stiff gradient for the cell to overcome. If on the other hand, we're using active transport and group translocation, for active transport, you only move 10 by active transport, and the gradient then would be, instead of 1 over 19, it will be 1 over 9, meaning there's 1 outside the cell let's just see if I can. No, I can't do that easily. Excuse me.
Bless you.
Thank you. So by active transport, we have 1 outside the cell and 9 inside the cell. So the gradient the cell is moving against is 1 over 9. And then the other 10 move by translocation. Wasn't sure I could fit the by in there. And that also the last one to move in will be 1 outside the cell, moving inside. Oop. And it's not 9 glucose molecules, it's 9 glucose 6 phosphate. I'm going to just abbreviate phosphate. So once again, the gradient is 1 over 9, which is a much easier gradient to overcome than 1 over 19. Any question about any of that? I don't use chat. I'm not sure what that means, but oops, that should be a T there. Anyways, that's why the prokaryote cell, can use active transport and group translocation. And then half the molecules can move into the cell by active transport, and the other half can move into the cell by group translocation. And because the group translocation is changing the molecule, we then have a less steep gradient than we would if we were only moving the molecule into the cell by active transport alone. Any question about the advantage of group translocation? All right, let's move on to talking about the cytoplasm. The cytoplasm is the substance inside the cell membrane. It's mostly water, about 80%. It does have dissolved solutes in it. It has the nucleoid region where the DNA is dissolved in it. It has ribosomes and inclusion bodies, which are not dissolved in the water or the cytoplasm. These are big enough, the ribosome and the inclusions, that they are not in solution. The inclusions can actually precipitate out of solution. The nuclear area, called the nucleoid region in a prokaryote cell, is wherever the single chromosome or DNA is floating in the cytoplasm. There is no distinction between the cytoplasm and the nucleoid region because there's no boundary. It's wherever the chromosome starts to float. There's no nuclear membrane separating the nucleoid region from the cytoplasm. It does cause the cell to stain differently, what's called the nucleoid region. We can also have some plasmid short molecules of DNA external to the chromosome, and they're not part of the nucleoid region. Or at least they don't have to be. Here we're looking at ribosomes, little dots in the cytoplasm. Ribosomes are big enough that they do not go into solution. Remember, ribosomes are made up of rRNA, ribosomal RNA, and then ribosomal proteins. They're the site where proteins are made in the cell. You don't need to know that the ribosomes have different units that make up the ribosome, but you do need to know that the subunits are made of protein, ribosomal proteins, and ribosomal RNA. You should also know that prokaryote ribosomes are a little smaller than eukaryote ribosomes. Prokaryote ribosomes are 70S in size. Eukaryote ribosomes are a little larger at 80S in size. The S stands for the sedimentation coefficient, and that's where the ribosome will sediment on a ultracentrifuge gradient. That's how biochemists and molecular biologists separate and isolate ribosomes. They spin it on a ultracentrifuge gradient, and then the prokaryote ribosomes will be in the 70S region of the ultracentrifuge gradient. You do need to know that there are different types of inclusions. An inclusion is simply a reserve deposit within the cytoplasm that the cell is storing. And so many molecules of what the cell is storing accumulate that the area precipitates out of solution. We have different types of inclusions. That's all you need to know. You don't need to know the different types. So the cell, which let's say it is a sulfur granule inclusion, the cell is accumulating sulfur inside the cell. So much sulfur accumulates in one area of the cell that the sulfur actually precipitates out of solution. Once it does, you can then see the inclusion. When it's before it is precipitated out of in solution, you do not see the inclusion because it's dissolved in the cytoplasm. But the cell, if it wants to use the sulfur, can start storing it, and the cell will store enough sulfur in the cell that the sulfur will actually come together, precipitate out of solution, and then form the inclusion. Any question about any of that? Here we're looking at some different inclusions. You don't need to know the types. Magnetosomes. There's the sulfur granules I talked about earlier. And this is a metachromatic granule. You can see the cell highlighted in the yellow rim there. And there's three inclusions in the cell. Any question about inclusions? If not, let's move and talk about endospores. Endospores are a specialized resting cell produced by certain species, mainly members of the family Bacillus and Clostridium. I think there's two other families, but both of the other families only have one species in the family. But Bacillus and Clostridium have many species in the two families. So the endospore is a resting cell that can survive harsh environmental solutions that will kill the parent or vegetative cell. So we have here, let me blow this up. We have here, right there, is a vegetative cell. It's not making an inclusion. Same with that one, not inclusion in endospore. Same with that one there. But that one is starting to make an endospore, and that's what we're seeing right there. Sporogenesis, also called sporulation, is the endospore formation. So these cells are going through sporulation. So is this one. And then germination is where the endospore returns to the vegetative state, where the cell no longer is an endospore, it's become the parent or vegetative cell. Let's walk through endospore formation. When you have endospore forming in the cell, it's because the cell is finding a deteriorating environment, and the cell will know that the parent cell will eventually die in this deteriorating environment. So the parent cell begins to make an endospore. What the cell will do is first, it will separate its DNA, replicate its DNA, sending one copy to one side of the cell, the other copy to the other side of the cell. And then the cell will begin dividing in two, but it doesn't split down the middle. It makes a smaller portion and a larger portion. The cell will then grow a plasma membrane in between the two DNA molecules, forming a smaller cell and a larger cell. The larger cell continues to develop the endospore, the smaller cell, and so we do continue this and call it the parent cell, which at one point was the one cell. Right here would be one cell, which then divides the nucleus into two. So this one will be called the parent cell. It remains a vegetative cell. And then this cell will actually grow a double membrane. So there's two membranes around this cell. You can notice the cell moves a little bit inside the membrane. It depends on what bacteria species it is. Some of the endospores stay at the very edge of the cell, so they do not move. Others move inwards a little bit. Others move inwards all the way, so they move in halfway. It just depends on the species of bacteria. All right, and then in between the double layer of the plasma membrane, meaning there's two plasma membranes literally inside this cell, you put down a peptidoglycan layer in between the two membranes. You then encode this cell with a very thick spore coat, the coating of the endospore, and still the endospore is growing within the parent cell, which is this cell here, until eventually environment becomes detrimental enough that the parent cell dies, and then the endospore can be released from the parent cell. Any question about any of that? I do have a little video on that, but it's not the best. See if it loads. There it goes. Sort of. It's taking its time. I'm not sure why it's so slow. So
Grammarly now uses AI to help you
after
write,
the
not just
commercial,
revise. Type a prompt and generate a persuasive.
so let's start at the very beginning again. There's a cell. It starts forming an endospore. All right. Any questions about any of that? I think before I begin the next slide, I'm going to just stop here a little bit early, and we will continue with our next slide next time. Any questions? All right, if there's no questions, I'll see you in the lab at 6:30. Bye.